My name is Jesper Olsen, and I'm the director of the Novo Nordisk Foundation Center for Protein Research here at University of Copenhagen, where I'm running a group in mass spectrometry-based proteomics. In my group, we focus on the mass spec technology development and applications to cell signaling networks, and particularly with focus in cancer research. We look at both peptides and their modifications, and we also now particularly focus on single-cell proteomics using very sensitive sample preparation workflows. When I think about the proteome, I think about the active players in the cell and those that actually do the work and make the cell the identity that it has. I think it's obvious that proteomics is really needed. Transcriptomics and genomics has brought us a long way, but it also shown that it's only 5% of the, for example, mutations that can actually explain a specific cancer phenotype. With the introduction of the thermoscientific Orbitrap Astral mass spectrometer, has really been a game changer for us in the lab. It has allowed us now to do things that we couldn't do before. It's basically 10 times more sensitive and 10 times faster in the acquisition compared to the previous generation of instruments. And this has allowed us to now move from cell populations to single-cell analysis. The Orbitrap Astral, we get much higher throughput now. So for example, we can analyze yeast proteomes routinely now with more than 100 samples per day and very accurately quantify all the expressed proteins in single runs. In our lab, traditionally, we've been a DDA lab, so data-dependent acquisition, and we only 5 years ago moved to data-independent acquisition. But now with the Orbitrap Astral, we can see that we basically get a merger of these two methods. So DIA originally was with wide window acquisitions, where you would typically go maybe with 10 or 20 Thomson windows across the whole mass range, whereas now with the Orbitrap Astral basically allowed us to go down to just 2 Thomson windows and cover the whole mass range. So essentially has changed the acquisition, and this of course gives a lot of advantages, so we get a lot more specificity in the DIA analysis. The thermoscientific Orbitrap Astral Zoom mass spectrometer will be a game changer because compared to the Orbitrap Astral, it is faster and is more sensitive and will allow us to do 300 proteomes per day. The main difference between the Orbitrap Astral Zoom and the Orbitrap Astral in terms of data size and quality is that especially for short gradients, we can see almost a doubling in the number of peptides and proteins that we can identify. One of the advantages of the new Orbitrap Astral Zoom is that due to the pre-accumulation and the new peak picking algorithm, we actually see more peaks. We can identify more fragment ions in each of the MS-MS spectra. With the Orbitrap Astral Zoom, we can now routinely do 300 human proteomes per day. This allows us to do very systematic systems biology type studies where we get sufficient coverage of close to 8,000 proteins and more than 100,000 peptides across all the samples. So we can do more replica, we can do more time, concentrations, experiments, and get more systematic across covering many different drugs and growth factors in the experiments that we record. We'll be able to identify the cell types, the most likely oncogenes that are activated, and the signaling pathways that determine the cell fate. With the Orbitrap Astral Zoom mass spectrometer will allow us now to study very small cells from, for example, from PBMCs, including T cells and B cells that have previously been extremely difficult for single-cell proteomics to analyze, but will now be something that we'll be able to do quite routinely with the enhanced sensitivity. The combination of the pre-accumulation, the faster acquisition, and the low input mode allow us to approach close to 7,000 proteins now in single-cell experiments. We get specific new information from especially the single-cell acquisitions using the Orbitrap Astral Zoom mass spectrometer, where we can now routinely detect and quantify very low abundant transcription factors, for example, stem cells that we couldn't do before. So hybrid DIA is an intelligent acquisition method where we try to combine both PRM, so parallel reaction monitoring, with the DIA acquisition strategies. So we've used hybrid DIA in my lab mainly to monitor very low abundant signaling pathways in, for example, single spheroids, cancer spheroids, that we have treated with different chemotherapeutic drugs. And using this strategy, we could actually detect phosphorylation sites that were not detectable by the standard DIA methods. My dreams for the field of proteomics, especially in the foreseeable future, is that we will be able to take the single-cell proteomics workflows to the next level, be able to analyze post-translational modifications in single cells, and also increase throughput from the current a few hundred cells per day to maybe thousands of cells per day routinely. The features of the Orbitrap Astral Zoom will help us to achieve this vision by enabling faster scanning, higher sensitivity, especially for these very low input workflows.